Okay, I think we're ready. I'm going to invite on the floor Professor Enza Maria Valente from Mondino Neurological Institute of Pavia and Martina Di Rocco from the National Institute of Health. They are going to chair this session. So thank you, and I'll see you in a bit.
Excellent. So thank you very much, Massimiliano, for the afternoon session that we are glad to chair along with Martina. We will have 6 talks this afternoon. We'll have 4 before the coffee break, and then you'll have a little bit of time to recovery, and then 2 more talks after the coffee break. So I think we can get started. We'll have 20 minutes for each talk, plus 5 minutes discussion, and we look forward to this session. Martina will introduce the first speaker.
We can start with Dr. Pirolli. It's a pleasure to introduce him for me. He's a researcher at the Rome section of the Institute of Chemical Sciences and Technologies of the CNR. And his research activity mainly focuses in the design of novel bioactive molecules as drugs candidates using computer chemistry techniques and artificial intelligence approaches. So, yeah.
Thank you for the kind attention. Thank you for the introduction. And before I start the presentation, I would like to thank the organizers for inviting me and presenting, of course, the project, and also a couple of data that we have produced in the last weeks. The project has been made possible by the Association Famiglie GNAO and HENI that gave us the financial and infrastructural support. Here? No, it's okay. This is the correct slide. Financial and infrastructural support. This project that has been just initiated started on March and sees the collaboration between Consiglio Nazionale delle Ricerche and any company. Actually, the title of the project is slightly different from the title you have read on the agenda of the conference, but the content is the same as it regards the structural study of the G-alpha protein that is encoded by the GNAO1 gene in order to assess the effect of the mutations on the structure and dynamic of the proteins, and with the final goal to assess if the G-alpha protein is a viable target for small molecules in the treatment of mutation-associated neurodevelopmental diseases, and of course, how this target can be druggable. For now, we took into an account 15 known mutations of this gene that causes several symptoms that can be divided into 3 groups on the basis of the clinical manifestations. The first group is related to disordered movements and is represented on the left column. Then we have the developmental and epileptic encephalopathy causing a mutation that is in the column on the right. And then we have 2 mutations that causes a hybrid situation between the 2 spectrums of symptoms. From a preliminary analysis of the crystallographic structures of the experimentally resolved structure in the protein data bank, we found that no direct interaction is in regard this mutation, no direct interaction with the interaction partners of G-alpha protein. So the idea was to use molecular dynamic simulations to assess what the mutation does to the structure and the dynamics of the protein. So we imagine that every mutation has an allosteric effects on other parts of the protein. For this, we imagined 5 milestones that we have planned to reach. We want to start, we have started because the first milestone is already finished, it's completed. We started from the structural characterization of the G-alpha protein in complex with GDP and GTP. We predicted the three-dimensional structures of the protein, the full-length human G-alpha protein complex with the 2 molecules. Then, after this, there will be the characterization of what these mutations will make to the G-alpha protein and dynamics, the protein structure and dynamics. Through molecular dynamic simulations. Then the information from the first 2 milestones will be used for the identification of a hot region on the G-alpha protein surface to be targeted with small molecules. The identification of small molecules that can bind tightly the surface, the hot regions on the surface of the G-alpha surface will be identified by virtual screening techniques. Final validation will be we will assess the ability of these ligands to revert the structural or dynamical effects of these ligands on the G-alpha protein. Given these milestones, we imagine the structure, the project structured into 3 work package, in which the first work package 0, regards the molecular modeling, so the generation of the 3-dimensional structure of the G-alpha protein and its variants in complex with GDP and GTP. The second work package 1, will regard the molecular dynamic simulations that will help us to understand how the mutations affects the structure and dynamic or essential motion of the G-alpha protein. After this, there will be the third work package, which is work package 2, which is the virtual screening strategy aimed to find small molecules that can bind the G-alpha protein, that can target G-alpha protein, and revert the effects, the dynamical effects of the mutations. The candidates found by this third work package will be then in a future project, will be also available for all to be tested with in vitro experiments. For what concerns the work package 0, the molecular models of the G-alpha protein and mutants have been already generated. We started from the APO form of the G-alpha wild-type protein because in literature, in the databases, there are 24 experimentally determined structure, but these structures are not complete, and not all these structures are complete. No human G-alpha protein is resolved in complex with substrate or the GDP. So we started, we modeled the APO structure of the G-alpha, and then we modeled the GDP and the GTP inside these proteins. For what concerns technical stuff of this process, we built the molecular model of the apoform by using a newly developed tool that is based on artificial intelligence, and we could build the 3D structure with high reliability. Then we use another newly developed tool that we can say is able to transplant the cofactors or substrates from non-crystallographic structure to your model. So we use this to generate the G-alpha GTP and G-alpha GTP bound forms. Okay. Well, starting from the two wild-type models, we built the other, all the variant listed that I mentioned before. So finally, we had 32 structural models of the G-alpha and its variants bound to GTP and GTP. For what concerned the work package 1, the molecular dynamics simulations, we just started a couple of weeks ago by simulating the wild-type G-alpha bound to GDP and the variant E246K. But this is only a preliminary simulation because we have to make other 3 replicas. So the data I will show I underline that, have to be confirmed. What we saw, basically, we saw that upon mutation, a structural, secondary structural element appeared. This one. I don't have the pointer, so sorry. I have 2 data, but appeared between residue 160 and 180. And another secondary structural element, in particular a beta strand, is stabilized when the glutamate 246 is mutated into a lysine. For what concerns the molecular dynamics simulation, is it possible to use it to understand how the protein binds, in this case, the GDP. And we, also in this case, we noticed some differences, in particular, how the serine 47 can bind the oxygens of the phosphate groups. I repeat, there are preliminary results, we have to confirm them. Another interesting application of the molecular dynamics is represented by the statistical analysis of protein motion, which is called principal component analysis. Principal component analysis is very useful to reduce the dimensionality of the high dimensionality of the protein motion. This data reduction, this dimensionality reduction is useful to capture the essential motions, the motions or the movements of the protein that are responsible, often responsible of the protein function. So, from the analysis of the principal components, we notice that if you see the first plot on the left, we notice that there is a reduction of the essential space explored by the protein when the E246 is mutated in lysine. So we can think about a reduction of the protein flexibility. Of the protein flexibility upon mutation. Thank you. Principal component analysis can be used also to assess which region of the protein is responsible for this difference in protein movements. In this case, the region, I don't have the point, the region between 270 and 300 undergoes to a great reduction of the movements. This is for the first principal component of motion, and also it is valid also for the second principal component. For what concerns the work package, the final work package, which is the work package that potentially will lead us to the final goal, which is to design molecules for the treatment of the disease, mutation-associated diseases. The virtual screening strategies, as represented schematically in a figure, and we want to start from a virtual library of 2 million compounds. Actually, we started, because it's just started this work package. We started from 2 million compounds, and we selected only those compounds that were able to pass the blood-brain barrier. We obtained about 100,000 compounds, and they will be docked. They will be submitted to docking-based prioritization that will say which of them can bind more tightly our protein. And the most stable of them will be further validated by molecular dynamic simulation that will help us to understand if the ligand is stable on the protein and if the ligand can give some changes in the mutated protein to revert the effect of the mutation itself. What's next? In the first 6 months, we are planning to produce all the data, all the simulations, all the virtual screening about all the 15 mutations. This is a lot of data, but we have the power with us. We have the power of the supercomputer from ANI that is a very useful tool in this case. So in these 6 months, we can generate all the data, and then we will pass to the analysis of the data. We want to first analyze a subset of mutations. We identified in the position 209, which is interesting, and position 246, because these two positions have two residues that interact with each other in the wild type. Then we want to study also the R209C, because it is the same position but give the different symptoms. And then we want to analyze the mutation G203R. Well, in conclusion, until now we generated 32 structural models of the G-alpha-O and its variants by the use of artificial intelligence. And these variants have been submitted to MD simulations, preliminary MD simulations. We built a library of molecules that for virtual screening that are specific for the central nervous system. And we noticed that principal component analysis of motion has emerged as a useful tool. I noticed an error. Has emerged as a useful tool to reveal the differences in protein motion upon mutation. In fact, the idea, and it can be verified, the idea is to use the output from principal component analysis to have a fingerprint of each class of the mutations and as an indicator of an indicator of one or other spectrum of symptoms. What we expect from this work, for sure, we are trying to reach the final goal, so to design a drug candidate that can modulate the protein function of the mutated proteins. Then we are going to generate a lot of data that can be a useful source of information about molecular basis of physiological effects of mutation on the GNAO1G. And also we hope that this data will be useful will be a guideline for mutagenesis experiment that can help us to study the relationship between structure and function of the G-alpha-O protein. The work will be carried on in the molecular modeling and virtual screening lab of the CNR, which is hosted in the Catholic University of Rome, and by the person of Maria Cristina De Rosa, which is the director of the Rome section of the SciTech, the CNR Institute, by me and Benedetta Righino, that are researchers, and hopefully by the voluntary help of PhD students and a master student. Aside this, we have also Silvia Pavoni and Francesco Frigerio from the R&D section of ENI that will give us a very, very useful help, especially in the analysis of the MD simulation. All these calculations will be carried out in the supercomputer from ENI, which is the most powerful supercomputer in the world, belonging to a company, non-governmental supercomputer, and is also one of the most green supercomputer in the world. So I finished my presentation, and thanks you for the kind attention.
Thank you very much. This is really exciting to see how technology comes into research and pushes research forward. Are there any questions for Davide from the audience? Can I start with, we have one there, so mine comes later. Please. Come.
Thank you for the presentation. I was just wondering, like with the E246 lysine mutation, right, which gives a movement disorder phenotype, then you find alterations in a region between 270 and 280 or 290, which is known for DEE development and less for movement disorder phenotype. So could you explain that, or do you have any hypothesis underlying that?
Actually, we didn't make any hypothesis because we have this preliminary data. So as I said before, this preliminary data have to be confirmed. The importance of the molecular dynamics simulations is that they can be used to cluster the mutations on the basis of the protein motion. To say, okay, that protein moves like this, and that mutations made the protein move like this, and can give these kind of symptoms. So for now, we didn't do any hypothesis about. Also because I repeat, there are preliminary data. Yes.
Thank you for the presentation. I'm very much in favor of applying this molecular dynamics to study the mutants, but I would like to ask a challenging perhaps question. So I have seen a work that studied GFP, green fluorescent protein mutants, and tested how well the alpha fold is capable of predicting the structures in these mutants, and basically it failed miserably. And the explanation is because alpha fold has been trained on the existing structures, and the mutations bring structures which AlphaFold didn't have to as the basis to be trained. So can you comment on this applicability of the AlphaFold approach?
Thank you for the question. It's my fault because I did not specify that I didn't use AlphaFold for building the mutants. I used a homology modeling approach. Actually, this is a good question. AlphaFold sometimes, especially in the disordered proteins, fails. If I have to see the truth, I didn't believe in AlphaFold. But in this case, and I'm also a computational chemist, so I cannot say this. AlphaFold works very well when you have templates with a high sequence identity with our models, with our sequence to model. And this was the case of the G-alpha protein. For what concerns the mutation, I agree with you. For these reasons, we decided to apply molecular dynamics simulation to investigate the structure also of the protein upon mutations.
So did crystallography failed in obtaining the structure of this protein?
Good question. I don't know, but crystallography, there are many crystallographic structure of the G-alpha protein, mainly in complex the GPCR, and we have also a structure of the G-alpha, but not human G-alpha, in complex with an analog of the GDP, an analog of the GTP. In fact, we use this structure to transplant the cofactors. Yes, the ideal situation is that it could be very, very useful to resolve crystallographically. Yes, I agree with you, yes.
Are there any other questions for Davide? May I just ask one last thing? It's very curious that sometimes you have very close mutations that you think they affect the same domain, and then the phenotype can be so completely different. You know, we see this in this disorder, but also in other disorders. Maybe one amino acid distance, and you have an entirely different impact. So how, from a biochemical point of view, you know, how can you explain this?
I can explain that this is an issue that has been investigated, because in proteins you can modify one amino acid and nothing happens, and you can modify the adjacent amino acid and all the stuff changes. Maybe the protein has not been produced by by the cell. This peculiar case have to be investigated.
Yeah, it's very important to model each specific mutation. Okay, no other questions? My co-chair, no questions? Okay. So, thank you again, Davide.
I leave the...
Come down.